Welcome everyone. Thank you for joining us today. We'll just give it a few minutes for everyone to filter in before we introduce the speakers and get started. So the Physiology of Obesity Mechanisms to Medicine series started a few years ago when myself and co-organizer Dr. Joe Lewis at the University of Cambridge felt there was scope to develop an ECR-focused symposium which could give more speaking opportunities to ECRs and bring those of us in the obesity and metabolic disease field together to network and disseminate results. After 3 successful symposia at the European Congress on Obesity, Experimental Biology, and the Day at Physiology 19, we've been delighted to bring this into a webinar series to give fellow ECRs the opportunity to present and network during a time when conferences are being cancelled and opportunities to do so are minimal. And with over 2,000 registrations across over 40 countries and another 2,000 plus views on YouTube, it's been great to see so many of you here as we begin the Nutritional Physiology webinar. I'll now hand over to Joe for some housekeeping rules and to introduce the speakers.
Thank you, Pete. Today's session will be recorded and will subsequently be uploaded onto YouTube for you to subsequently view there. If you'd like to submit a question during the talks today, please use the Q&A button at the bottom of the screen. Both speakers will give 20-minute talks and then we'll proceed to the Q&A session. Our first speaker today is Sofia Cienfeugos from the University of Illinois in Chicago, who will be speaking about 4- and 6-hour time-restricted feeding and its impact on body weight. Thank you, Sofia.
Thank you so much. Can you hear me? Yes. Okay. Thank you so much for inviting me today. I'm really excited to be here. My name is Sofia Cienfeugos. I'm a PhD candidate at the University of Illinois in Chicago. I work with Dr. Krista Verity, and we run human trials on intermittent fasting. So today I'm going to talk to you about the effects of time-restricted feeding on weight and cardiometabolic health. And I'm also going to talk to you about a study that we recently published on cell metabolism a couple of months ago. So what is intermittent fasting? I think that's one of the big questions. Everybody seems to be an expert on this topic nowadays. So there are different types of intermittent fasting that's been studied. The first one that's been studied is called alternate day fasting. So alternate day fasting is one of Krista's main area of research. Alternate day fasting is a form or a type of intermittent fasting that involves alternating fasting days and feasting days. So every other day of the week you would have to fast, and that fast could be a 0-calorie fast, so nothing, or it could be a very low-calorie diet between 500 and 600 calories. And then the other days you will eat your normal diet. Then we have another type of intermittent fasting that's called the 5-2 diet. It's similar to alternate day fasting, but you just have to fast for 2 days a week, and those are 2 non-consecutive days a week. And it's also a very low-calorie diet, so 500 or 600 calories. And the third type of intermittent fasting that's been studied, and it's the one that I'm going to talk to you about in this presentation, is called time-restricted feeding. And time-restricted feeding is different from the 2 previous types of intermittent fasting because it doesn't involve doing it a couple days a week or restricting calories. It's basically shortening the eating window every day of the week. So we all eat on a certain eating window from the first thing that we eat each day till the last thing that we eat on that particular day. So if we tend to shorten that period to less than 12 hours, we will be doing some sort of time-restricted feeding. Usually time-restricted feeding eating window ranges between 4 hours and 12 hours. And something else that's different from time-restricted feeding compared to the other 2 types of intermittent fasting is that it doesn't involve any sort of conscious caloric restriction. You don't have to count calories, you don't have to do any specific type of diet, any specific composition of macronutrients. You just have to shorten the time and eat your normal diet during those hours. So one of the most typical types or forms of time-restricted feeding is called the 16-8. The general public talks a lot about this one. So basically, during the 16-8, you prolong your nighttime fasting to 16 hours and you eat just on an 8-hour window. Usually that 8-hour window goes from 10 a.m. till 6 p.m. or it could go from noon till 8 as well. So there's a lot of books about intermittent fasting, a lot of YouTubers, a lot of experts, a lot of media, but actually we have very, very few human trials on intermittent fasting. We have more in mice models, in animal models, but in humans we have very, very few. We also have a lot of reviews, but human trials very little. So one of the big questions is time-restricted feeding effective for weight loss in individuals with obesity? Because it doesn't require any sort of caloric restrictions, just looking at the clock. So some people wonder what happens if someone just eats more than their caloric requirements on those hours. Are they going to still lose weight? Are they going to gain weight? So I'm going to talk to you a little bit about some studies. This is a study that we did in our lab a couple of months ago. No, sorry, a couple of years ago. And this was the first study analyzing the 16-8. So eating on an 8-hour window. So the intervention group, we had 23 participants that they started eating at 10 a.m., stopped at 6 and fasted from 6 p.m. till 10 of the next day. And we had a control group that continued with their usual diet without any time restrictions. We assessed different things both at baseline and at week 12, body weight, adherence, body composition, metabolic disease, among others. And we saw that there was a significant weight loss without any caloric restriction, any intentional caloric restriction on those 12 weeks. So the intervention group lost on average 2.6% of their initial body weight on 12 weeks. The control group remained the same. However, it's important to notice here that even though this was statistically significant weight loss, this is not clinically significant weight loss. So clinically significant weight loss is usually when you lose more than 5% of your initial body weight. So here it's just 2.6%. But still, it's interesting because there was no caloric restriction, no diet advice. And also here we can see that participants during the 8-hour window reduced their energy intake by 350 calories a day. So there was an unconscious caloric restriction of 350 calories a day. It's also interesting to see that their macronutrient composition didn't change significantly from baseline to week 12, but the overall calories did change. So the composition of the diet remained the same, but they ate less calories of that specific type of diet that they followed. So one of the most interesting things about time-restricted feeding, at least from my point of view, is that you don't need to monitor what you're eating. You don't need to select specific types of foods. You don't need to count calories. You just watch the clock, you just look at the time, and you will be unconsciously restricting calories. And this is something great because you just have to give one advice to people. Eat from here till here, and that's it. So we were wondering, we saw that with an 8-hour window, there was weight loss, but it wasn't clinically significant weight loss. And we also realized that by shortening the eating window, participants restricted calories without even realizing that. So we thought, what if we shorten that window even further? So if we shorten that window even further, are we going to maybe be able to achieve clinically significant weight loss? Or are we going to see better results in terms of weight loss or cardiometabolic disease risk factors or not? Is it the shorter the better? So we decided to run the first randomized clinical trial with short time-restricted feeding windows. So we had 4-hour time-restricted feeding group. So participants started eating at 3 p.m., stopped at 7 p.m., and then fasted from 7 till 3 of the next day. The 6-hour time-restricted feeding group, they started at 1 p.m., stopped at 7 p.m., and then fasted from 7 till 1 of the next day. And the control group, they continued with their usual diet without any timing restrictions. They did this for 8 weeks, and we assessed as well body weight, body composition, adherence, metabolic disease risk factors, sleep, both at baseline and at week 8. So so this slide, I put this here because there's a lot of people that ask, how do you know the participants were actually eating or that they were actually doing the intervention? So we, of course, this was self-reported. So we gave this log to participants every week. So they had to fill the date, at what time they started eating, and at what time they stopped eating. And I would fill these parts according to the time that they started or stopped, if they were adherent or not. And we also gave participants a 7-day food record, both at baseline and also at week 8. They had to write everything that they ate on 7 days a week. So we assessed their energy intake, their diet composition, diet quality, macronutrient composition, etc. So this is the subject flow chart. We screened 82 participants. 24 were excluded from the because they didn't meet one or more inclusion criteria. However, no subjects declined to participate. That was also surprising. 58 participants were randomized to one of these 3 groups. 19 participants were randomized to the 4-hour group. 20 participants were randomized to the 6-hour group, and 19 participants were randomized to the control. We had some dropouts, but no dropouts due to disliking the diet. Most of the dropouts were due to scheduling conflicts or unable to contact, but we had no dropouts due to, like, I'm not being able to follow the thing. We had 16 completers on the 4-hour group, 19 completers on the 6-hour group, and 14 completers on the control group. So these are the results for weight loss. After 8 weeks of both 4-hour and 6-hour time-restricted feeding windows, we saw that both intervention groups lost on average 3.2% of their initial body weight. It's also interesting to see that both interventions lost the same. There wasn't any differences between the 2 groups. In the beginning, we hypothesized that the 4-hour was going to lose or was going to achieve better weight loss compared to the 6-hour, but we actually did not see that. So at least according to this study, fasting for longer than 18 hours wouldn't induce any extra benefits, at least in terms of weight loss. It's also interesting to see that this wasn't a clinically significant weight loss. It was statistically significant again, but not clinically significant. However, the curves looking down, maybe if this study was a little bit longer, we could have achieved clinically significant weight loss, but we don't know that. These are the results for the adherence. We calculated this based on the log that I showed you a couple slides before. And personally, this is one of the findings that surprised me the most in the beginning when we were about to run this study. And when I knew that I had to make people eat on just 4 hours, I was really scared. And I thought that no one was going to be able to do this and that everybody was going to dislike the intervention and that they would not adhere at all. And I'm really, really impressed that they were actually able to adhere on average 6.2 days a week, so almost every day of the week, to both interventions. And the adherence remained high throughout the 8 weeks of the study, of the trial. And we also had no dropouts due to disliking the diet, so impressive. These are the results for glucoregulatory factors. So after 8 weeks of 4-hour and 6-hour time-restricted feeding, we didn't see any effects on fasting glucose, but we did saw significant decreases on both fasting insulin and insulin resistance on both intervention groups. We saw that insulin went up on the control group. We can discuss that later because that's a little bit surprising, but on both intervention groups, it went down significantly. Here are some of the metabolic markers that did not change. So we didn't see any effects on blood pressure, heart rate, and we also did not see any effects on LDL cholesterol, HDL cholesterol, or triglycerides. We also were wondering the effect of time-restricted feeding on inflammatory markers and oxidative stress. We didn't see any effects on inflammatory markers such as interleukin-6 and TNF-alpha, but we were surprised to see that there was a significant decrease in 8-isoprostane. So this is a marker for oxidative stress, and both intervention groups decreased these markers significantly after 8 weeks. Also, this is some of the results that surprised me the most, and I think this is one of the most interesting things about time-restricted feeding. Again, the unconscious caloric restriction. So participants in this study without counting, I'm going to repeat this 1,000 times, without counting calories, without looking at what they were eating, without selecting specific foods, they restricted on average 550 calories, both intervention groups. So a lot of people wonder, is time-restricted feeding safe? Aren't you going to induce some severe adverse events from eating in such short eating windows? So we also measured adverse events during the entire 8 weeks of the intervention. It's interesting to see that dizziness, headache, nausea, and diarrhea were increased during the first week of the intervention. So the first week that they actually did the fasting. So on the first week, you might experience some of these adverse events. However, after the first week, they tend to disappear and they weren't significantly different from controls. So this is similar to other forms of weight loss strategies. At the first week, participants tend to feel bad because their body's sort of getting used to this new way of eating. And then they tend to disappear. We didn't see any effects on fatigue or irritability. We did see a significant increase in constipation on the first few weeks of the intervention. However, the one that persisted, most of these adverse events disappeared completely, but dry mouth was the only one that persisted throughout the trial. And participants reported getting used to it in the end. However, we spoke with some odontologists and experts, and they mentioned that saliva production is also dependent on chewing things. And during the fasting hours, they are basically allowed to drink non-caloric beverages, but they are not able to eat things. So the absence of chewing or eating solid things does not stimulate the saliva production. So that's one of the explanations why this happens. Another question is, won't I be too hungry to sleep? Isn't this going to affect my sleeping outcomes? So we also measured sleep. We didn't see any effects on sleep quality or sleep apnea risk. So fasting doesn't affect negatively or better sleep quality or sleep apnea, but we saw a significant decrease on insomnia severity on the 6-hour time-restricted feeding group. This was surprising. However, this is self-reported data. We didn't do any polysomnogram or more sophisticated measurements. So I think this is something worth studying even further in upcoming trials. So summary of these findings, there was a 3% weight loss in 2 months, and it was the same for both interventions. So the 4-hour didn't induce more weight loss. So both groups decreased the same amount of weight in 2 months. Also, self-reported adherence was high in both intervention groups. And there was an unintentional caloric restriction of 550 calories a day. I think that this is one of the most interesting findings, because making someone restrict calories of 550 calories consciously, so doing a low-calorie diet that's 550 calories below their usual diet, usually has very, very poor adherence. So this one had high adherence. It's also interesting to see that we saw significant decreases in both insulin and insulin resistance in both groups. So this would be a good strategy for people with insulin resistance or pre-diabetes. Also, oxidative stress decreased significantly on both intervention groups. However, we just measured one marker of oxidative stress. It would have been better to assess more markers. Maybe for upcoming studies, that would be a strategy. But we didn't see any effects on lipids, inflammation, or blood pressure. Some practical considerations: who should not do intermittent fasting? I think this is really important to mention. Pregnant women shouldn't do intermittent fasting, and pregnant women shouldn't do anything that induces a negative caloric balance. Lactating women shouldn't do intermittent fasting because we don't have any studies about the safety of intermittent fasting or lactating women. Shift workers also shouldn't. They are also usually excluded from these type of trials because they're awake at night and sleep during the day, so the circadian clock and everything is disrupted. People with strict medication regimens that require ingesting food with medication also shouldn't do intermittent time-restricted feeding or intermittent fasting. Also, people with any substantial health condition shouldn't do intermittent fasting by themselves. They always should talk with their primary care physician. And it's also important to mention that intermittent fasting is not for everyone. There's such a big hype about intermittent fasting nowadays that people tend to feel that they should be doing and that everybody should be doing intermittent fasting. That's like it's so important for your health, and it's not like that. Intermittent fasting is an alternative, but it is not something that everybody should do it. Some people are going to feel great. Some people are going to feel terrible. We still don't have enough data to know what's the profile of subjects that do better with intermittent fasting or worse. We don't know that yet. So some people are going to do great, some people won't. So acknowledgements to my lab mates, Mark, Faisa, Kelsey, Serbi, Krista, my boss there, Shuhao, Manuela, and Vicky, and also to all the great participants that were part of my study. Thank you very much for listening.
Thank you, Sophia, for that interesting talk. We'll proceed to our second talk now from Lauren Milley from the National Institute of Health, who will talk to us today about ad lib energy intake from both plant-based low-fat diets versus animal-based keto diets. Thank you, Lauren.
All right, can everybody hear me?
Yes, thank you.
Okay, thanks so much for having me today. Let me get my videos out of the way. Okay, so my name is Lauren Milley. I'm a postbac research assistant working in Dr. Kevin Hall's lab at the National Institutes of Health, which is located right outside of Washington, D.C. Today, I'll be talking about the results of our most recent clinical trial that we wrapped up just this past March. A plant-based, low-fat diet decreases ad lib energy intake compared to an animal-based ketogenic diet, an inpatient randomized controlled trial. So a little bit of background, like Sophia was talking about, one of the major issues is a rise in obesity, which is due to a change in the food environment, probably driven by excess caloric intake. So there are a lot of theories about why calorie intake has increased, but if it's as simple as too many calories in, a simple solution presents itself. We can restrict calories, but that raises more questions like what kind of calories? So I'll be focusing on macronutrients. What kind of calories should we be restricting? What kind should we be eating? Two major hypotheses come up about this, and one of them is the carbohydrate insulin model of obesity, and this really paints carbohydrates as the bad guys. So Harvard physician David Ludwig and his colleague Kara Epling, who are also at Harvard, explain this pretty well by saying a high carbohydrate diet produces postprandial hyperinsulinemia, promotes deposition of calories in fat cells instead of oxidation in lean tissues, and thereby predisposes to weight gain through increased hunger, slowing metabolic rate, or both. So you kind of enter this vicious cycle when you consume carbohydrates because you get this high insulin response, which puts the body into storage mode. So you get storage of lipids and a decrease of circulating metabolic fuels, which then makes you feel hungry and leads you to just eat more, all at the same time while decreasing your energy expenditure. Contrast this with the passive overconsumption model of obesity. So you can see when you look at calorie density, what 500 calories looks like. This will tell you that eating high fat foods, which are high in energy density, are going to be your enemy, because when you eat 500 calories, it doesn't really fill up your stomach. You can see in the oil picture that you still have an empty stomach. You're still going to feel like you need to eat more, so you're going to passively overconsume calories. Fruits and vegetables are going to be good things because they have low energy density, they take up large volume in the stomach, and so you feel full more quickly. So a summary of these hypotheses are that the carb insulin model predicts that a high glycemic, low-fat diet is going to lead to postprandial hyperinsulinemia, promoting fat accumulation, and then leading to excess caloric intake. The passive overconsumption model predicts that a high-energy, low-carb diet is going to lead to excess caloric intake and weight gain. So which one is it? That's what we wanted to find out with the primary aims of our study, which were to determine differences in ad lib energy intake in kilocalories during 2 weeks of eating an animal-based low-carb diet as compared to 2 weeks of a plant-based low-fat diet, when matched for presented calories and protein. Secondarily, we also wanted to determine the differences in ad lib energy intake just during the final week of each diet. And that was because we didn't have a run-in period and we didn't have a washout period either. So when we brought healthy volunteers into the clinical center, we didn't maintain weight stability beforehand, and we didn't have time in between the two diets, and so we wanted to eliminate any spillover effects that might cause. Here is a graphic that's showing the schedule of procedures over the 28 days that people stayed in the clinical center, which is the hospital on NIH's campus. So volunteers were randomized to either start on the low carb diet or the low fat diet, and they would spend 14 days on that one and then immediately switch for a total of 28 days. And over the 28 days, a battery of tests were performed, including fasting blood draws, DEXA scans, which stands for dual X-ray absorptiometry, and from that we got body composition data, so we could see changes in fat composition, fat-free mass, bone mineral density over time. We did mixed meal tests, oral glucose tolerance tests, and we also have 24-hour chambers we have 3 metabolic chambers where we can determine through indirect calorimetry free living energy expenditure. So patients spent 24 hours in those chambers once per week. Here's some information on our subjects. So the inclusion criteria included males or females. We had 20 total volunteers, 9 females, 11 males, aged 18 to 50. The average for our volunteers was about 30 years old. They had to be weight stable and with a BMI of greater than 20 kilograms per meter squared. Our average was about 28, so slightly into the overweight category, but pretty representative of the general population, with an absolute body weight of greater than 53 kilograms. And they were also required to be able to complete stationary cycling for 20 minutes during the screening process. And we did clamp exercise at 20 minutes 3 times a day on a stationary bike, and that was just to replicate the kind of exercise that people would have, mild to moderate exercise during free living, because we know people kind of tend to slow down when they bring them into the hospital and have them stay on a hospital unit for 4 weeks. We also did this to prevent things like blood clots. Some of the more common exclusion criteria were hypertension, so people with high blood pressure were not able to participate in the study, and people with low hematocrit levels. So these are the diet compositions. Our animal-based low-carb diet was made up of 75% fat, 10% carbohydrates, and 15% protein. And 82% of that energy that was presented to patients was made of animal products. So most of the calories are coming from meat, poultry, fish, eggs, dairy, and nuts. The plant-based low-fat diet reverses the carbohydrate and fat percentages for 75% carbohydrates, 10% fat, and again holding protein constant at 15%. And 100% of the energy on this diet was made from plant products. So that was a lot of root vegetables and soy products, whole grains, and fruits. One of the goals of this study was really to test the gold standard of each diet. So we want to give the keto diet, low carb diet, its best fighting chance against the best plant-based low fat diet that we could create. So both diets were gonna contain the same base of non-starchy leafy green vegetables like lettuce, peppers, asparagus, and Brussels sprouts, and all of those good things that both diets usually recommend. To give you a little perspective of what those percentages are like compared to diets that you might be more familiar with, I have a graph here of carbohydrate percentage on the y-axis and protein percentage on the x-axis. And the standard American diet, at least, is composed of about 50% carbohydrates and 15% protein. For all the people in the UK, I'm not sure if that's different, but you might be familiar with the Mediterranean or the paleo diets, which tend to limit carbs to some extent, and those are between about 20 to 40% carbohydrates. But ketogenic diets, and at least a well-formulated ketogenic diet, is going to be seen in this blue circle at the bottom, which is much lower than any of the other diets listed, at less than 15% carbohydrates and less than 20% protein. And so our ketogenic diet is well within that space, right in the middle at 10% carbs and 15% protein. The Ornish diet is listed in purple at about 65 to 70% carbohydrates, and that was originally created to improve cardiovascular health. And so our vegan diet, our low-fat vegan diet, is even above that at, again, 15% protein and 75% carbohydrate. So this is what the menus actually look like. So these are meals that the patients consumed on the low-carb diet. You can see cheesy eggs for breakfast, steak salad for lunch, and important to note are the sides. So we had a lot of ranch dressing, guacamole, sour cream, and using cheese and dressings were one way to get the fat content as high as we needed it to be. And also for dinner, this patient had zoodles. So anytime we tried to give them pasta, it was going to be a vegetable substitute like a zucchini noodle. This is an example of the plant-based diet, and this is blueberry quinoa for breakfast, just a regular pasta type salad for lunch, and then a lot of rice, vegetables, and fruits for dinner. And something that's not evident from this picture is this, the serving size. So these glass bowls are huge. They're like what you would use to serve a family of 4, and this is all for one person for one meal. So you can contrast that with the size of the breakfast on the low-carb diet as compared to the low-fat diet. The volume of food they were getting on the low-fat diet is just much, much larger. So we have a quick validity check. Like I said, we wanted to test the energy density hypothesis and contrast that with the carb insulin hypothesis. So we want to make sure that our low-carb diet is actually significantly more dense in terms of kilocalories per gram than the low-fat diet. And it is. So on average, volunteers were presented with about 3,900 calories per day, and the density of the low-carb diet was 2.1 kilocalories per gram, compared with the low-fat diet at 1.1 kilocalories per gram. When you look at non-beverage energy density, that's even a little bit higher on the low-carb at 2.2 versus 1.1 again on the low-fat diet. We also saw the kind of insulin response that we wanted to see from the low-fat diet, which caused pretty high excursion following a meal, and this data is taken from the average of patients after a mixed meal tolerance test where they were given a shake that had the same composition as the diets. So at the end of the plant-based low-fat diet, they were given a shake that was composed of 75% carbs, 10%, 15%, and we do see a large insulin response, and we don't see that on the low-carb diet, which is what we wanted. So here's the first data slide. This is the average energy intake averaged across our 20 patients over the 14 days on each diet. The animal-based low-carb diet in red and the plant-based low-fat diet in green. And what you can see here is that the low-carb diet on every single day across all 14 days led to a pretty significantly higher energy intake in terms of kilocalories. So something that you might get when you're averaging across your patients is to average out outliers. And you can see that we have a few at the top that in terms of absolute number of kilocalories definitely ate more than the average, but everybody was following the same trend. So all 20 patients, each one is represented by one of these lines, was consuming more on the low-carb diet as compared to the low-fat diet. And this was on average a magnitude of about 700 kilocalories per day. So the average of the low-carb diet was 2,700 kilocalories per day as compared to the low-fat diet, which was about 2,000 kilocalories per day. We also want to make sure that what these people are consuming is what we're presenting. We don't want patients on the fat diet to be able to pick out the cheeses or not be using the dressings and not actually be consuming this 75% fat. So when we do our weighbacks and we do our analysis, we do see that they're consuming very close to what's presented. The only significant difference is protein intake. So on the low-carb diet, volunteers consumed 1.5% more protein than on the low-fat diet, and in absolute terms, that was about 130 kilocalories per day more in protein on the low-carb diet. This was statistically significant, however, it's not a huge difference, so I would venture to say that this 1.5% difference in protein intake isn't enough to explain the large magnitude of difference in energy intake between the two diets. We also measured their appetite and their eating rate, so we did a lot of surveys before and after each meal, and there was no reported difference on a visual analog scale from 0 to 100 in pleasantness or familiarity, or hunger, satisfaction, fullness, and eating capacity. So I thought that this was surprising, that people seem to be similarly familiar, they seem to like the diets the same amount, and they had really no difference in how full they felt after either one. When we measured eating rate in terms of grams per minute, so when we're looking at volume, it's no surprise that the plant-based low-fat diet, there was just a lot more volume there, led to a quicker eating rate. When you measure in terms of density or in kilocalories per minute, then you get the reverse trend, and the low-carb diet is going to lead to faster intake. You can also see that when we look at the data in terms of each meal, so when we separate it out by breakfast, lunch, dinner, and snacks, it's pretty consistent that the low-carb diet led to greater energy intake across all meals. So it wasn't all concentrated at a single meal like breakfast. Every single meal that was eaten, there was more eaten on the low-carb diet as compared to the low-fat diet. So now we have the weekly energy intake comparison. Like I said, we had that second primary aim that was just going to be looking at the second week of each diet. And you can see that that makes a significant difference for the animal-based low-carb diet. When you look at the first week compared to the second week, people were really slowing down by the second week of eating the ketogenic diet. And that was by about 300 kilocalories per day. So that's a pretty significant drop-off, whereas there's no difference in the first and second weeks on the plant-based low-fat diet. However, even with the drop-off on the low-carb diet, it's still going to be higher than the plant-based low-fat diet. And one of the reasons or mechanistic explanations for this might be ketone levels. So one of the big things with the ketogenic diet is that ketones, which are a byproduct of your fat oxidation, are appetite suppressant. So we wanted to make sure that we had a long enough period of stay that ketone levels would rise to the point of appetite suppression. And by measuring ketones, and we have an N of 15 for ketone measurements, we do see that after a week, those ketones are rising and then kind of leveling off. There is somewhat of an upward trend after day 7, but it's pretty stable. And you also see that in the energy intake trend, while it does level off concurrently after about 7 days, that trend is also going back up. So it's hard to say what would happen if we had kept people in for longer, but it does seem like there was enough time for that adaptation. So looking at the average change in body weight, both diets led to a weight loss. The animal-based low-carb diet led to more weight loss at an average of 1.7 kilograms compared to the low-fat diet at just about 1 kilogram. So that was surprising to see that people were eating 2 to 3, some people were eating 5,000 calories per day, but still getting weight loss on both diets. Both of those were significant from baseline, but they were not significant compared to each other. So what was that weight loss made of? For the low-carb diet, the majority of that weight loss was coming from fat-free mass. So after 14 days, participants on the low-carb diet were losing about 1.6 kilograms, and we think that that's mostly water weight and glycogen, whereas on the low-fat diet, there was basically no change in the fat-free mass. So that's going to be opposite for the change in fat mass. On the low-carb diet, there's really no change in fat mass. We actually saw a very, very slight increase after the first week, and then a decrease after the second week. But no significant change after either weeks from baseline. The low-fat diet was opposite, where we saw a decrease in fat mass after both the first week and the second week. However, when you compare the two diets after the 14 days, we have a p-value of 0.085, so statistically that wasn't a significant difference between the two. However, if you look in terms of the rate of fat loss, that is going to lead to a significant difference with a p-value of 0.019. So in the low-carb diet, it was about a loss of 16 grams per day compared to the low-fat diet, which was about 51 grams per day. So here's some data from the metabolic chambers on energy expenditure. You do see that on the low-carb diet, energy expenditure is going to be higher, and when you break that down into the sleeping and sedentary energy expenditure, it's higher on the low-carb compared to the low-fat diet. Physical activity expenditure, however, was basically the exact same. And again, we did limit participants' exercise to 20 minutes 3 times a day on the stationary bike, and our metabolic chambers have toilets, and they have the stationary bike, and they have TVs, and patients are allowed to bring in their belongings. So we do our best to make this as much of a free living experience while being in the chamber as possible. The respiratory quotient for the low-carb diet was about 0.75, which is to be expected when you're burning mostly fats as the metabolic fuel source, and it was about 0.88, 0.89 on the plant-based low-fat diet, which is consistent with burning carbohydrates. So here's some data on the continuous glucose monitoring. To be expected, we used the continuous glucose monitors weren't on the hip, and those were kept on for all 28 days, and the plant-based low-fat diet led to higher continuous glucose, or average glucose at about 94.3 compared to the low-carb diet at about 81, and the coefficient of variation was also higher at about 18.4% as compared to 13.5%. Both of those still in a healthy range though. We can also look just after meals, so just the postprandial effect of glucose, and you can see that the low-fat diet leads to a much higher excursion. You get the increase and then the slow decrease, whereas the low-carb diet, there's not much change. These are the results of the mixed meal test. So C-peptide is also going to correspond with insulin release, and as expected, when on the low-fat, high-carb diet, you get higher C-peptide levels than the low-carb diet. The same trend is going to be true for lactate. Low-fat was higher at all time points than low-carb. Triglycerides were somewhat interesting because we know that low fasting levels of triglycerides are a good thing, and the low carb diet did seem to lower fasting triglycerides, but the postprandial excursion was so much higher that actually the average triglyceride level on the low carb diet ended up being higher than the low fat diet. Free fatty acids followed the pattern that was also expected. The low carb diet led to higher levels of free fatty acids than the low fat diet. Here are the results of the oral glucose tolerance test, and these were, for the most part, also in line with what we expected. The only thing that I would call your attention to is the glucose levels. So on the animal-based low-carb diet, when taking oral glucose tolerance test, we know is used for diagnosis of diabetes, you have slight impairment on the low-carb diet. But remember, these are healthy volunteers. None of them have prediabetes or diabetes. And in terms of diagnosis, the adaptation to a low-carb diet might be somewhat misleading during OGTTs. So in conclusion, the carb insulin hypothesis predicted that the high glycemic low fat diet would lead to postprandial hyperinsulinemia and thereby promote fat accumulation and excess intake. But we saw that the plant-based low fat diet actually led to fat loss and lower intake. So we're going to say that this one is mostly false. The passive overconsumption model predicted high energy density animal-based low carb diet would lead to excess caloric intake and weight gain. And so we did see that the low carb diet did lead to higher intake, but it also would have led to weight loss. So this one is, I think I'm frozen here. Okay, half true. I think the biggest takeaway though is both of these models are probably too simplistic to explain weight loss, body fat, mass loss, and fat-free mass loss. So we're gonna have to go back to the drawing board. And there's also a lot of heterogeneity between patients. So I don't think that there's anything conclusive for either of these at the end of the day. I'd like to thank my PI, Dr. Hall, and all of the people in my lab, also all of the people, the nurses, the clinical center, the metabolic kitchen. It's not easy to keep 20 people in the hospital for 28 days straight and to feed all of them. So thanks to this very big team of people that we have at the NIH. Thank you very much.
Thank you, Lauren. Another excellent talk there. We've had lots of questions coming in from the Q&A button. Thank you for those. I'll just kick us off while a few more questions come in. Sophia, if I could start with you, please. I'd be really interested to know, does the timing of the time-restricted feeding window matter?
So apparently it does. So we have a little bit of data showing that early time-restricted feeding might be better for in terms of metabolic disease. So there's been a couple studies comparing early versus late time-restricted feeding, most of those done by Dr. Courtney Peterson. And they've seen that apparently earlier is better. However, in our study, the time that we used would be considered late for American standards. But because we were doing such short eating windows, we were really scared of adherence, that people were going to drop out. So we wanted to enhance adherence as much as we could. And we knew from previous data that people, the most important meal for them to eat with their families is dinner. And if you don't allow them to eat dinner with their families, the probability that they were going to stay in the trial was really low. So we tried to put the eating window somewhere that was like easier for people to follow rather than metabolically better. So in future studies, maybe it would be a good idea to put it a little bit earlier and see how that works.
And a kind of follow-on question I had from there is, did you measure circulating hormones, particularly after the first few weeks of the intervention?
Circulating hormones?
Yeah, after the first few weeks of your intervention?
We did not.
No, okay. Because leading on from there, one of the questions we had was, in both yours and the initial 8-hour time-restricted feeding window, there's rapid weight loss in those first few weeks before it kind of stabilizes. And did you have any thoughts as to why that might be?
This is something that's very common in most weight loss. Usually most weight loss graphs, independent of the strategy that you use, the first week of weight loss is the biggest one because you also lose a lot of water there. So there's a lot of water weight there, and then it tends to stabilize. But usually the first few weeks is where you see the fastest weight loss. Yeah, but it's usually more due to water loss in the beginning a little bit.
Craig Sale was interested to know what happened to the levels of physical activity in your intervention groups.
Yes,
could you comment on those, please?
Yes, we measured, participants had to wear like a Fitbit. So we measured physical activity and steps, both at baseline, in the middle of the intervention, at the end. We told participants to stay doing the same amount of physical activity that they were used to. Don't change, don't increase, don't do anything different. And the amount of physical activity was the same between groups. So there wasn't a group that was doing more than the other. However, it would be definitely interesting for upcoming studies to include physical activity, because some studies in time-restricted feeding that include physical activity has shown better lean mass preservation.
Thanks for that, Sophia. I'm just going to have a look through the rest of the questions. So there's a question: were the participants allowed to drink water during fasting or chew gum? I think you answered one of them. I think they're allowed to drink.
They were allowed to, yeah, they were allowed to drink water and any non-caloric beverages, so tea, coffee, herbal, yeah. But gum, I told participants to avoid gum, because usually gum has some sort of calories, and sometimes gum can induce hunger because you're secreting, or like gastric acidity, because you're secreting gastric juices while you're chewing. So sometimes it makes people feel like acid reflux. So I told them, like, try to avoid, and to the general public, try to avoid gum during fasting hours. Yeah.
Great. And then there was on that topic again, there was a question from Luis Salazar, who asked whether the participants all consumed similar amounts of coffee or tea during the fast, and whether consumption of antioxidants through these drinks could have had any of that impact on oxidative stress.
Good, really good question. We actually didn't count, didn't compare the amount of tea or coffee that they consumed during their fasting. We did not do that, and I think that's a really, really good idea for. Maybe we can do it. No, we didn't do that, but that's a really good idea. Yeah.
Great.
Yes,
there was also a question from Kenneth Verboven asking how applicable is time-restricted feeding in type 2 diabetic patients, and the risk of hypoglycemia, etc.
Yes. So for type 2 diabetes, the only type of intermittent fasting that's been studied is the 5-2 diet that I mentioned in the beginning. So there's a big study, a good study that was done in Australia with the 5-2 and type 2 diabetics, and it showed some really good results. They were comparable to daily caloric restriction, but they were closely followed by their endocrinologist, because the medication regimen has to be changed, because there's a lot of risk of hypoglycemia if you continue with the same amount of medication. However, we don't have any studies in time-restricted feeding and type 2 diabetes. So I wouldn't advise to do time-restricted feeding in people, so people with type 2 diabetes to do time-restricted feeding, unless they talk with their physician and they try a specific scheme, like that, a short, maybe not too long of a fast, like maybe a 14-hour fast, or maybe 12 hours or 14 hours, maybe, but we don't have data yet, so I prefer to say no.
Okay, great. And then, so I think maybe we could probably bring Lauren in on this question. So I wanted to know, so how does the macronutrient profile of the eating days affect the impact of the intermittent fast? Do you know if there's any difference if on those days they eat maybe like a high carb or a low carb diet, for instance? Does it matter?
That question is for me, right?
Yeah, but I thought with Lauren's talk, maybe she might have some thoughts on that as well.
So at least from our study, there wasn't any changes in their macronutrient composition at any point of the study, at least on the times that we measured their 7-day food record. The diet composition was the same, similar from what happened with the first study that I mentioned. The diet, like the amount of carbs, the amount of protein, the amount of fat remained the same. Just the overall calories was the thing that changed. So we couldn't assess if maybe a higher carb or a lower carb would induce some changes, because none of those things changed.
Okay. Yeah, so I think that was my question. In like the free living population, do you think do you think the fasting days would be enough to induce benefits regardless what the rest of the diet was like on the eating days?
So if the fasting was enough to achieve those results?
Yeah, regardless of what the diet was like on the eating days, do you think the fasting would be beneficial?
That's what we saw in our study, that it actually, something that I forgot to mention is that diet quality in the beginning of our study was pretty bad, pretty low. And at the end of the study, it's still pretty bad, pretty low. The only thing that changed was the overall calories. But we saw significant results in terms of insulin, in terms of oxidative stress, in terms of weight loss. So apparently fasting in itself has an effect. Now, if we combine that with a healthier type of diet, with a Mediterranean diet, with a low carb, with a low fat, I don't know if we have better results. We don't know that yet because we don't have any studies modifying what people eat during those hours. That's something that we want to do, but we don't have that.
Great, thank you. Joe, I think you had a couple of questions as well.
Yeah, Lauren, some of our attendees were interested. Could you confirm the kind of weight range of your population and also their BMI ranges as well?
Yeah, so we had a really large range. We actually had some participants that were less than 20 in the beginning, and that was why we actually put in that greater than 20 kilograms per meter squared, because our lowest, I think, was about a BMI of 18, and that was a male. And we were seeing some more effects from hypoglycemia during the low carb diet on the very thin people. And the highest was probably in the high 30s. So there was quite a large range.
Yeah. And someone was interested in the percentage of protein within the two diets. Can you confirm what that was, please?
Yeah, so that was held constant at 15% for both diets.
And does that, I'm sorry, I forgive my naivety here, does that reflect kind of your average individual's protein intake over your standard meal?
Yeah, I'm not sure at the range of individual intake, but for the most part, it seemed like people were not kind of picking apart diet. So whatever we presented them with, they were eating it as served. So I wouldn't expect that there would be a large difference.
Yeah, and I think you had this on one of your slides, but I didn't quite fully take it in at the time. And did you have any data about the fiber consumption in your participants?
So there was much higher fiber content in the plant-based diet, and that was kind of, I think, inescapable.
Yeah. Were those the ones with the lower food intake, if I can remember correctly, or the lower energy intake overall?
Sorry, can you repeat the question?
So the fiber intake was greater in the plant-based diet. Yeah, okay, that's fine. Thank you very much. There was another question here. Hold on while I just find it as I'm discussing this. This one again was from Craig Sale. And given the impact of muscle glycogen levels and hydration status on DEXA body mass composition outcomes, how were the pre-scan conditions controlled in your experiment, Lauren?
So we did the DEXA every single morning at 8 a.m. after a 12-hour fast. So they were allowed to drink water, but typically they didn't. We told them to stay in bed the morning of. So there was usually at least an 8-hour fast with no drinking or eating prior to that.
Okay, thank you. Pete, can we switch back to you for a minute while I just organize myself here?
Yeah, no worries. I'm just going through the questions. So I think this is back for Sophia now. So Tangang Aluang wanted to know if there was any association between A-type isoprostane and body weight, or I guess any other parameters, if you've looked at that.
We didn't actually run the stats for that, but usually most of these markers are usually related with weight loss. And that's one of the big questions in fasting. So one of the big questions is, are these results because people lost weight, or are these results because of those fasting hours? And that's a question that we don't have the data yet to tell you with 100% certainty which one is. From the data that we have nowadays, I'm afraid to say that most of the benefits are due to weight loss. But there's been a couple studies, actually one study that was done in the UK that was a great study. It was a short study, but it was like a very mechanistic study that they were analyzing the effects of fasting on skeletal muscle metabolism. And they saw that compared to a caloric restriction matched in everything, calories, macronutrients, everything, the group that was doing the fast, their skeletal muscle had a better capacity of glucose and amino acid uptake compared to those that were doing a normal caloric restriction. So maybe there is something that's just because of the fasting, but we're not 100% sure yet how that works. So, but yes, insulin resistance, maybe oxidative stress, and all of those metabolic benefits that we've seen in the previous studies are usually related to weight loss. So,
great, thanks for that. That answered a couple of the other questions about whether it was due to the fast or the reduction in calorie intake. And I guess the important thing is, regardless of the means, it's about the calorie reduction and the weight loss. And
exactly.
Seems like a great tool for some to do it.
So that's one of the important things I say, that time-restricted feeding is a great strategy for people that cannot restrict calories every day, for people that just can't change everything that they're eating, can't eat, I don't know, like cauliflower rice every day, or like change foods. Just by looking at the time, you will be restricting calories. So it's like an alternative for people that find normal daily caloric restriction difficult. So it's like an alternative.
And just to follow up from there, and this is a question from both of you. So if we start with you, Lauren, please. Did you do any follow-ups with your participants to see if the weight loss was maintained?
No, we did not.
Okay. Sophia, did you either?
Unfortunately, we did not. That would have been great, but we did not.
Yeah, I was particularly interested with your participants, given that they had this unintentional caloric restriction of around 600 calories, to see if they did maintain that when they were kind of released from the study.
We don't know. This is just anecdotal data. I know a couple participants remained in contact with me and told me that they kept on doing it and kept on losing weight. But this is not something that we measure. This is just anecdotal. But I'm afraid that because of the high adherence and the no dropouts, so we had high adherence and no dropouts. So I think that maybe people continued doing it because they weren't sick. So most of the students were happy doing it. They were excited. They were like, I'm still eating whatever I want to eat and I'm still losing weight. So I'm afraid maybe they continue doing it, but this is just what I think. We didn't measure it.
Yeah, it was interesting. I think you had more dropouts in your control arm than your intervention arm, which I found amusing. But
the control group didn't do any. So the most difficult thing for me when running this study was keeping the controls in the study, because the control groups weren't doing anything. They weren't doing the diet, and we didn't pay participants. So we were like please stay. And of course, some of them just dropped, and the one that stayed, I did the intervention to them at the end. So I did the time restricted feed. I did like nutrition counseling for them. So the ones that stayed was because of that, because they didn't have any reason to stay in the study, honestly.
Did either of you make any further measurements on loss of muscle and bone mass with either of your time restricted or your kind of intervention on dietary macronutrient preference?
Yeah, I can start. So since we have the DEXA scans every single week for patients, we do have that. We didn't record it. It wasn't one of the secondary aims of our trial, so that's not something that was listed in the paper or published ever, but we do have that data. It wasn't that long. It was 2 weeks per diet, and we were keeping an exercise at least constant at a moderate level. So I wouldn't expect to see a huge loss of bone mineral density or bone mass, but that is something that we have looked into.
Yeah, we didn't saw any changes in bone density. We didn't publish that, but we did see a decrease on muscle mass on the 6-hour group, and this is something that was really strange, because if we were like, it would have been more normal to see muscle mass loss on the 4-hour group, not the 6-hour group. So the 4-hour group were maintain their muscle mass, and the 6-hour group lost muscle. We asked a lot of experts on the field, and all of them said there's no explanation for this. However, there's been other studies in time-restricted feeding that have seen muscle mass loss, and those studies are the ones with ad libitum diet like ours and without exercise. So my recommendation when I tell the general public what to do when they do time-restricted feeding is that they need to pay attention to the amount of proteins that they're eating, and they need to do some sort of physical activity in order to preserve lean mass. Definitely.
Great, thank you guys. Thanks for 2 fantastic talks, and there's been some great engagement, lots of questions today. So I'm going to close here, and hopefully we'll see you all back next week. So next Monday we have our final Physiology of Obesity session. This time it's a Meet the Expert session with Professors Julian Zirath and Mark Reitman, and I hope to see you all there. Thanks everyone.
Thank you for inviting us. Thank you.
Bye.
Bye.